From the Schaefer collaterals to the terminal, a larger amount of glutamate is released from the presynaptic terminals. This is comparable to use studying for longer periods. This results in higher frequency action potentials. When glutamate binds to the AMPA receptor, a greater depolarization event occurs. The AMPA receptors remain open longer due to the increased concentration of glutamate, which allows a larger amount of sodium to enter through the AMPA receptors. This influx of sodium causes a large depolarization event in the postsynaptic cells, which repels the magnesium blockade from the NMDA receptors through a process known as electrostatic repulsion. At this point, the NMDA receptor, with glutamate bound, allows sodium and calcium to enter through its pore. In this way, NMDA receptors are known as coincidence detectors because they require a presynaptic and postsynaptic event for channel opening. The binding of presynaptically released glutamate and a significant postsynaptic depolarization via activation of the AMPA receptors. As previously mentioned, LTP is the process of strengthening the connections between two neurons. The influx of postsynaptic calcium acts as an important secondary messenger, activating many secondary intracellular cascades. The increase in calcium contributes to two phases of LTP, early phase and late phase. During the early phase, the calcium binds to its respective binding proteins and causes the insertion of new AMPA receptors onto the postsynaptic cell membrane at the active CA3-CA1 synapse. These AMPA receptors are stored in the postsynaptic CA1 internal cell stores and will only insert when there is a large influx of calcium through the NMDA receptor. This allows more AMPA receptors to be available for future depolarization events. The early phase changes last for only a few hours and require a brief increase in calcium levels. During the late phase, a prolonged influx of calcium causes an increase in transcription factors, ultimately resulting in gene expression and new proteins to be synthesized. Some proteins resulting from this process include AMPA receptors, which are inserted into the postsynaptic cell membrane at the synapse. In addition, there is an increase in the synthesis of proteins called growth factors. These are involved in the formation of new synapses, which is the basis for